talk will be 10 plus 5 minutes, if I am correct, and Johnny Stibian will be presenting a mitochondrial complex 1 deficiency disorder related to protein, nucleotide binding protein-like. So floor is yours, and time starts.
Thank you. Thank you. I would like to thank the organizers for allowing me to present my work here. And I will be discussing mitochondrial disease, mitochondrial complex 1 deficiency disorder related to a protein, another protein, NUBPL. We all know complex 1, NADH dehydrogenase, NADH ubiquinone oxidoreductase is the largest of the electron transport system. And it's made up of 45 protein subunits with very important prosthetic groups, the FMN, and 8 to 10 iron sulfur clusters that are absolutely required for the electron transport capacity of the protein. So these iron sulfur clusters need to be incorporated into the protein. And here I'm discussing the nucleotide binding protein-like, or NUBPL, initially called iron sulfur cluster transfer protein, specifically for NADH dehydrogenase. In 2009, it was discovered that this protein, deficiency in this protein leads to severely dysfunctional mitochondria, and therefore it was absolutely required for the assembly and maturation of complex 1. Yesterday, very nicely, Nick Lane explained to us that iron sulfur clusters can originate spontaneously. However, in mammals, there are other types of enzymatic reactions that are necessary for the incorporation of iron sulfur clusters. The mitochondrial iron sulfur cluster system and the cytosolic iron sulfur cluster system. Just to make a long story short, there are several proteins that are necessary for the incorporation of iron sulfur clusters. It starts by cysteine desulfurase that removes the sulfur from the cysteine into the core ISC complex using electron transfer from NADPH and ferredoxin reductase and ferredoxin. It transfers the electrons with the iron that is imported into the mitochondria, and this, using multiple chaperones, forms the 2 iron 2 sulfur cluster, which can be transported into proteins, or it can be transported outside of the mitochondria for the use in the CIA machinery and in iron homeostasis, or another iron can come in and other electrons coming in using the same system to make the 4 iron 4 sulfur clusters that can be transferred, can be imported into its own proteins, for example, by NUBPL for complex 1 or NFU1 for complex 2, etc. So NUBPL is an extremely important protein for the complex 1 health. And genetic defects in NUBPL lead to a subclass of primary mitochondrial disease. It's called complex 1 deficiency nuclear type 21 with a concomitant increase in complex 2 activity as a compensatory method. In HeLa cells, knockdown of NUBPL leads to mitochondrial remodeling of the respiratory complexes, leads to a loss of cristae membranes and increased lactate production because of over-reliance on fermentation. And of course, there is an absence of proper function of the electron transfer system. The first patients to be discovered with a compound where compound heterozygote having two different mutations in the NUBPL gene. The paternal allele was a missense mutation in the conserved glycine, glycine to arginine, whereas the maternal allele had an exon 7 duplication and a deletion in exons 1 to 4. This first patient had developmental delays, leukodystrophy, and elevated cerebrospinal fluid lactate due to complex 1 deficiency, and other mutations were observed and studied along the lines. So NUBPL, the disease, it leads to a deficiency in complex 1, and it is inherited as an autosomal recessive pattern. It's mapped to chromosome 14q12, and it has multiple neurodegenerative manifestations summarized in this table. Of course, since it is a mitochondrial disease, it affects mostly all tissues, and all tissues will have problems with this in this genetic disease, and multiple outcomes can come out. So this protein has a similar, it's similarly to cytosolic NTPases in the CIA pathway, and like them, it does not have the ability to coordinate iron sulfur cluster on its own. So it's likely a dimer because this dimer can incorporate the iron sulfur cluster to transfer it to other proteins. So we actually identified the Drosophila melanogaster homologue of this protein. It was essentially called CG3262 protein, and it shows stark homology to the human NUBPL. We cloned it, we overexpressed it, and we localized it to mitochondria in immunofluorescence and in Western blotting. And we purified this protein as a dimer, irrespective of its iron sulfur cluster status. So it has a Stokes radius of 37 angstrom, which is estimated molecular mass of 66, corresponding to the theoretical mass of 62 kilodaltons. So it is purified as a dimer. Very interestingly, when we overexpress this NUBPL homologue in Drosophila S2 cells, the results is starkly similar to overexpression of mitochondrial DNA helicase, whereby replication stalling is now removed. So it actually helps in mitochondrial DNA replication. So we also checked the membrane binding. Of course, this is specifically for NADH dehydrogenase, so it has to bind to membranes, and you can see here that using different types of assays, we were able to show that there's a nice binding depending on liposome concentration, and it does not depend on any cardiolipin, any specific lipid content of the liposomes, whereby the other protein, NTD, which is the N-terminal domain of the mitochondrial DNA helicase, shows a strong dependence on membrane binding on cardiolipin content. So both of these proteins bind to the mitochondrial inner membrane. And there has been a paper showing that these two proteins might be interacting. This is very interesting because the Drosophila form of NTD has, the Drosophila form of the helicase has an iron sulfur cluster, and it is possible that the NUBPL transfers the cluster to the helicase to induce the activity. So basically what I wanted to show you is that NUBPL is a devastating genetic disease characterized by problems in complex 1 and OXPHOS, and it may have implications on mitochondrial DNA replication, and the studies are actually ongoing in my lab. I want to also point that mitochondrial DNA helicase contains iron sulfur cluster in the N-terminal domain, and this is required for the stability of the protein. And the NTD binds to cardiolipin-rich liposomes, and NUBPL bind to membranes with no lipid specificity, and it could be a binding partner between NUBPL and the helicase to transfer iron sulfur cluster to enhance DNA replication. How is this related to the disease? How it is related to the human mitochondrial DNA helicase which has no iron sulfur cluster is currently under investigation in my lab. With that, I would like to acknowledge the great work by a former undergraduate in my lab, Muna Abed Rabbo, and the collaboration with Laurie Kaguni and Minyoung So at Michigan State University. Thank you very much.
Thank you. The perfect timing. So now we have time for questions. Are there any?
Great talk. I was wondering whether in the Drosophila mutant of NUBPL, you have a compensatory effect of dysfunction of complex 1 by bisphosphate beta-glutamate.
This is something that is currently under investigation. I don't have the right answer right now, but it is something that we are actually actively looking at. Thank you.
More questions?
It's not on.
From the epidemiological side, so iron deficiency is a very strong contributor to the global burden of disease, and it, of course, affects women more than men. And iron is also important for the synthesis of neurotransmitters like dopamine. And it is also relevant, so in Alzheimer's, for example, there is accumulation of iron in the brain causing neuroinflammation. Do you also see in your experiments any signs of inflammatory processes, and is there any kind of interplay?
We actually looked at in cells, basically. We did not move to an organism level, so I don't have an answer to your question, but I think this is something worth looking into.
Thank you. More questions from the audience? If no,
I cannot. Or you can? No,
Okay.
go ahead, please.
I was going to ask also about additional models to Drosophila because it's clearly very important clinically. So what do you see? Do you see any models further in maybe rodents or?
Well, mammals, for the mitochondrial DNA helicase in mammals, basically in rodents or in humans, we do not have iron sulfur clusters. So we are actually looking into that at the moment right now. We just want to see if there is any interaction between these two proteins, and if there is an interaction, what does it do to the protein? Does the overexpression help with the replication stalling or not? Because it's clearly shown in the Drosophila, but the Drosophila requires the iron sulfur cluster in the mitochondrial DNA helicase. We don't. Does it do any difference or not? We're still looking into that.
Any other questions? No. Then I will try. As you know, there are many mutations in complex 1. No? So then in the context of this perspective, this mutation that you are describing, how it relates in the assembling of the complex 1? Or in the functionality you already saw with the OSFO system, but it affects more to the M module or the assembling of the complex 1 and the proper functionality, or what is the main feature that difference from the other mutations?
So basically, this mutation is not in complex 1. So this is a mutation, it's in a different protein that is required for the assembly of complex 1. So basically, if this mutation is present, or these mutations, because there are multiple mutations in this protein, the complex 1 is actually formed. It is deformed. It's formed, but not in a correct way. So the iron sulfur clusters in complex 1 are needed for functionality of the enzyme, but they're also needed for structure of the enzyme. So the membrane arm, the matrix arm is a little bit deviating from the normal. Yeah.
Thank you. Any other question? Adam, last one, quick. Yeah.
Just a quick follow-up. Have you done any kind of super complex analysis on these to see? I'm curious. If
We did not, but other groups have started with this work by looking at super complexes. But this is something that is extremely important to look at and see what the whole thing looks like. Thank you. Thank you.
Okay. If there's no further question, we close. Please. Thank you very much.